The second question has to do with, why do you think RNA is used to classify microorganisms at this point? Well, let's really think about what RNA does. First of all, RNA is only single-stranded. Okay, remember that DNA is double-stranded. DNA is a more stable molecule. RNA is a much more variable molecule. It can be cut, it can be spliced and joined together. And what we're learning about in relationship to bacteria is how rapidly they can recombine and how easily they can change and become new strains based on things like antibiotic resistance. So if you think about the ancestral tree model, for instance, and say we have a particular bacterial species, let's say bacteria number 1, in the presence of antibiotics, we can have recombination of genetic material, and then we can have a strain, let's call it strain A, and then a strain B, and then based on the selective pressure, the natural selection that occurs with antibiotics, we could, for instance, select for this strain B. And one of the mechanisms that can actually distinguish these two strains from one another very uniquely is RNA, even more so than DNA, because potentially strain A and B could have DNA that's very similar or even the same, but the RNA will be potentially different because the strains are different. And so that way you can actually distinguish between the strains by using RNA. So as we go through, particularly in our discussion about bacteria, we're really going to talk about how these strains develop and what are some of the criteria that promote the development of these multiple strains. So that concludes our discussion about classification of organisms. Thank you so very much for listening to Educator.com.